Hello, everyone. So this is week 3, lecture 4, and we're going to be talking about mapping the mind or the brain in action. How do we know what we know about the brain and what it can do, right? This is actually a very recent field in which we've been able to really get inside our heads and see what the brain is doing and how different activities correspond to different brain regions. And scientists are developing ever more advanced technologies to let us map the brain. So let's take a quick history lesson. Back in week 1, we talked about pseudoscience, and I told you about phrenology, which was the first attempt to map the mind onto the brain. Phrenologists believed that bumps on the skull corresponded to brain enlargements, and that these brain enlargements were linked directly to psychological capacities. So they would literally feel a person's head for any bumps or enlargements, determine which area of the brain they corresponded to, and then make conclusions about the person's psychological makeup. Now, we know today that's not exactly scientifically rigorous, and so later, newer methods replaced phrenology. One of these methods is studying people with already damaged brains. So some famous examples are the studies of SM, who had complete bilateral amygdala damage, and the earlier study of Phineas Gage, who was the guy with the tamping iron through his frontal cortex. Scientists would examine the deficits that these people showed in order to assess what functions were critically associated with certain brain areas. Now, that way is still viable, and we still use that strategy of studying people with brain damage in order to see what's going on in different brain areas. But there's some newer methods as well, such as stereotaxic procedures. These are a little bit more precise. They allow scientists to pinpoint more specific areas. And neuropsychologists are able to rely on even more advanced tests, like measures of reasoning, attention, verbal and spatial abilities, and assessing cognitive strengths and weaknesses to determine where they correspond with different brain regions. Today's technology has provided even more advanced strategies for assessing brain function. So we're going to walk through each of these. I like this chart. I think it's a good organizational system for all of the different types of scans and their abbreviations because they can get a bit unwieldy. But we'll start out by talking about electrical stimulation with the EEG. We'll move on to talking about different brain scans and imaging techniques, and we'll end by talking about magnetic stimulation and recording. So let's dive in. An EEG, or an electroencephalograph, is a test that's used to evaluate the electrical activity in the brain. If you'll recall, we talked about how brain cells communicate through electrical impulses. An EEG can be used to detect any potential problems with this communication system. An EEG tracks and records brainwave patterns. That's what you see in the image all the way towards the right. So as you can see in the picture on the left, small flat metal discs called electrodes are attached to the skull with wires. These electrodes then analyze electrical impulses in the brain and send the signals to the computer where the results are recorded. And the electrical impulses in the EEG produce a chart that looks like wavy lines with peaks and valleys, sort of squiggles. These lines allow doctors to quickly assess any abnormal patterns. Irregularities may be a sign of seizures or brain disorders. EEG is a non-invasive method, and it's really good at detecting rapid changes in electrical activity, so it's pretty frequently used to examine brain functioning. So I want to show you a video of what an EEG looks like. Come on.
My name is Sam and I'm here today to have an EEG. Feeling a bit nervous as I've not had one before, but I'm sure it'll be fine. The technician, Fiona, explained what happens. She said the EEG records brain activity and it takes about 40 minutes. She measured my head and made marks with a pen so she would know where to put the electrodes. The 2 electrodes on my chest monitored my heart rate. The electrodes were fixed in place with paste and tape. It felt a bit scratchy. Fiona put them all over my head, but once they were there, I couldn't feel them. Once all the wires were hooked up, Fiona began recording. I laughed when she said the EEG doesn't read your thoughts. Fiona asked me if she could use some flashing lights during the test and open and close my eyes. Next, she asked me to breathe in and out slowly for a couple of minutes to see if this changed my brain activity. At the end, Fiona took off the electrodes and cleaned my hair with cotton wool and water. Doctors sometimes use EEGs along with other tests to look at your brain activity. Fiona told me my neurologist would go through my results at the next appointment.
A revolution in brain research occurred with the advent of brain scans, or neuroimaging, methods that allow us to peer inside the brain structure, function, and sometimes both. A CT, or computed tomography scan, uses a series of X-ray images taken from different angles, and then it uses computer processing to create a cross-sectional image. It sort of takes slices of the brain. Magnetic resonance imaging, or MRI, uses the release of energy from water and biological tissues after exposure to a magnetic field. These are better than CT scans for detecting soft tissues like brain tumors. I have a lot of videos in this one. I'm sorry that it's going to make me jump back and forth.
Hi, I'm Angie, and your doctor has ordered an MRI, and I'm here to show you exactly what the MRI machine looks like and what it does. What it does is a big camera that takes a picture of the inside of your body. How it works is you will lay on the table, and the bed will move up, and it will go in, and it will take pictures just like you would with a normal camera. It just looks a little bit different. Once you're in the camera, it doesn't hurt, it doesn't spin around on you, or it doesn't close in. All it does is make a really loud noise, and that noise does not hurt. It's just very loud. So a lot of the time, the kids will listen to music or put headphones on, and this is what these look like. And I want to show you on a little model. So this is kind of how the machine works. We shrunk it down to show you exactly that you will lay on the bed. There will be a place there for your head, and you will lay right there. And the bed does move, and it will move into the camera, but your whole body will not go all the way in. And it does not close. It does not have any doors. You can see all the way through it. This part of the camera also does not move. Nothing spins around. All it does is make really loud noises. And again, the ladies will let you wear headphones or listen to music. When you're in here, you will always hear a little bit of a sound. Some kids like to say that it sounds like a bird chirping or like a car humming, and that is normal. If the doctor has ordered to get a picture of your brain to look at the inside of your head, this is part of the camera also, and it will slide over your head. It kind of looks like an astronaut helmet, or it looks like a football helmet, and it does not touch you. It just goes on the outside of the pillow that you will already be laying in. And when you're lying there, there's a mirror here. So you will be able to see whoever is with you by just looking up with your eyes. We don't want you to move side to side or up and down. If you move while you're in the camera, it will become blurry just like a normal camera would. So you can look with your eyes and see whoever is standing out here so that you're not alone. And if you choose not to listen to music or to have the headphones in, they also might use little pillows like this, and they go right next to your head. They're nice and squishy, and that just makes you more comfy and also makes it harder for you to move your head. Because again, your job is to lay very still as you can, without moving your head around so the pictures are not blurry. And now we're going to show you exactly how the bed works and what sound that the camera makes so that you're not afraid of it.
Okay, I think you get the point. This is obviously a video to help people who are going to get an MRI not be frightened, but I think it does a good job of sort of showing you what the process looks like. CT and MRI scans can show only the brain's structure. It can't actually show activity. So for information on thought or emotion, we need to use functional imaging techniques. So unlike the CT and MRI scans, which can just look at the structure or the build of your brain, positron emission tomography or PET scans and functional MRIs or fMRIs allow us to measure both structure and function. So a PET scan measures consumption of glucose-like molecules to give a picture of neural activity. It measures changes in the brain's activity in response to a stimuli. It can be considered invasive as it involves an injection of radioactive molecules, but it's not long lasting and it's not harmful to our brains or to our bodies. The fMRI uses magnetic fields to visualize brain activity using changes in blood oxygen level. This provides detailed images of activity in small brain regions over brief time intervals. So it's considered to be an advantage over the PET scan, although it's very sensitive to motion. So this means you have to stay super still. Think about like if you were taking a picture with a real camera. If you're going to move your hand around a lot, the picture is going to be blurry. fMRI techniques are usually used to measure brain activity in response to specific tasks. So next I want to show you a PET scan.
A PET scan creates 3D images of the body. It does this by using radioactive tracers, which are usually administered to a patient through intravenous injection. The tracers are made up of carrier molecules that are tightly bonded to a radioactive atom called an isotope. The carrier molecule can interact with or bind to specific proteins or sugars in the body. The carrier molecule that will be used depends on what the doctor is looking for. If she suspects cancer or is monitoring a known cancer's growth, she may use FDG, a modified form of glucose, which gets absorbed by tissues. When tissues absorb a lot of glucose, it may indicate a cancerous tumor. The radiation from the tracers poses little danger to the patient since they quickly pass out of the body. The isotope produces small particles called positrons, which interact with surrounding electrons. This interaction results in the complete annihilation of both particles, releasing two photons that speed off in opposite directions. The detectors in the PET scanner measure these photons and use this information to create an image of the distribution of FDG in the body.
So let's move on now to talk about some other imaging techniques. Transcranial magnetic stimulation, or TMS, applies strong and quickly changing magnetic fields to the surface of the skull that can either enhance or interrupt brain function. Because the TMS is actually interfering with brain functioning, we can think of it as an independent variable, something that we're manipulating. Therefore, TMS is the only non-invasive brain imaging technique that allows us to infer causation. All the other techniques can only correlate brain activity with psychological processing, since they're not manipulating anything. There is some research to suggest that TMS can provide relief for depression and may decrease auditory hallucinations or hearing sounds that aren't there. So there is some evidence to say that repetitive TMS may even be a treatment for depression, but that's still very much up in the air and under investigation. Magnetoencephalography, or MEG, measures tiny magnetic fields generated by the brain. The strength of the MEG is that it's able to track brain changes over extremely small time intervals, in contrast to the PET and fMRI scans, which measure change from second to second. MEG measures activity changes by millisecond. So as I hope you've realized by now, we use different parts of our brain for different activities. Scientists refer to this as the localization of function, or identifying brain areas that are active during a specific psychological task over and above a baseline rate or activity. The media tends to oversimplify localization, so keep in mind that the brain is extraordinarily complex, and often complicated psychological functions are linked to multiple brain areas. Now, the cerebral cortex has two hemispheres which work very closely together and are connected by the corpus callosum. Generally speaking, each hemisphere is responsible for different functions. This is known as lateralization. So we know that the left hemisphere is primarily responsible for fine-tuned language skills, such as reading and writing, as well as actions such as facial expression and motion detection. The right hemisphere is primarily responsible for more coarse language skills like tone of voice, and also visuospatial skills like facial perception. But what happens if the corpus callosum, or that bridge between the two hemispheres, gets severed? This is known as split brain surgery. It's a very rare operation. It's really not done that much anymore, but it used to be a form of treatment to alleviate seizures. Studies of split brain patients reveal a bizarre fragmenting of mental functions. So this image here shows a split brain patient. She's conducting a task where she's shown a snow scene and a claw up at the top. And she's been asked to identify the matching object. So the right hemisphere of her brain is responsible for actions. So that sees the snow scene. As we'll talk next week, everything is opposite. So her right hemisphere looks onto the left side. That sees the snow scene and the shovel. So it results in her pointing with her left hand because the hemispheres control the opposite sides of the body. Now her left hemisphere, which is responsible for language, is going to see the claw working across on the right side, which will result in her saying chicken out loud. There's a lot going on there. We don't need to get into the nitty-gritty. The take-home point is that split brain patients often experience difficulties integrating information that's presented to their two separate hemispheres, since the two different sides are no longer speaking to one another. In our last section for week 3, we're going to talk about nature versus nurture, which is a really common and popular topic in the field of psychology. So I will see you all then.